Now, of course, we all look after patients with infections, and probably, like me, from time to time you look after people suffering from sepsis, from severe overwhelming infection and the immunological response to that infection that the body generates. But most of the people, most of the time, don't have infections. And the reason for that is that we are protected by the immune system. So the immune system is that which protects us against infection. We have immunity. We're protected against infection. And of course, this is so important in all fields of clinical practice, but so important for humanity. The most common cause of death in human history has been infection. People have died of infection, sometimes in childhood, sometimes in later life, sometimes in epidemics. The mind probably goes to smallpox in South America, documented to have killed at least 300 million people. That the mind goes to 1348 in this country, the year the Black Death, the bubonic death, arrived in this country. But how come most of the time most of us are protected? It is the study of the immune system. Now, there's two main types of immunity. There's that which is innate, and innate immunity is normally non-specific. And we have innate immunity via innate inbuilt systems in the body, but they are normally non-specific in nature. That means they act against a wide variety of potential infecting organisms. And then the other type of immunity is acquired immunity. And the acquired immunity is immunity we don't have at one stage, but then we get it, we develop it. And the difference here is that the acquired immunity is specific to a specific organism. So we're exposed to that organism and then we develop specific acquired immunity, as opposed to the innate immunity that works against a wide variety of organisms, whether or not we've encountered them in the past. So let's look at this in a little more detail now, and we'll start off with a mind map to explain the basis of immunity. So, just like that guy said, we can see that immunity can be divided into innate, which is the non-specific type of immunity, or acquired, which is the specific form of immunity. And then what it's useful to do is subdivide the forms that the innate immunity can take. So these are all different forms of innate immunity, these varieties here. Now, first of all, we think about normal anatomy and physiology. So normal anatomy and physiology incorporates protection from infection in the very design and structure of the body. And we're going to look at some of those in detail in a few minutes if you just hang on. But let's look at these other ones first. Now, the inflammatory response is innate. It's not specific to anything. Now, what I mean by that is the inflammatory response is the body's generic response to any tissue insult. So it doesn't really matter whether that tissue insult is burning or cold, or it's an immunological reaction, or whether it's a trauma, whether it's a cut, whether it's a bump, whatever it is, that the inflammatory response is always the same. And the inflammatory response gets white cells to the area. It gets immune proteins to the area to protect us against infection. And it's also the first stage of the healing process, which in turn is going to prevent further infection because we're going to heal the integrity of the tissue. Now, another generic response is phagocytosis. Phagocytosis. Now, phago means to do with eating, and cytosis, of course, is cells. So let's think about what's happening in this example of phagocytosis. So in phagocytosis, what we have here, let's do those in green. So we've got a couple of bacteria here have managed to get into the body. A couple of bacteria. And these bacteria have a kind of smell, for want of a better term. They release molecules, bacterial molecules, that indicate their presence. And that'll be dictated by a phagocytic cell. So here's a phagocytic cell. In this case, it's a macrophage in the tissues. Now, macrophages are a type of monocyte, and these are big, so they're sometimes called, well, macrophages are big eaters, that's what they're called. But there's also cells called microphages, which are the neutrophils, primarily the neutrophils, which are much smaller cells, but will also eat. So the phagocytic cells, and they detect these bacteria, and they will move towards them. And they will move towards them by what's called amoeboid movement. So they will be streaming in the cytoplasm, and they will move towards these bacteria. And the cell will end up getting close to these bacteria. So the cell's now close to the bacteria. But when it gets close, it doesn't stop, it just keeps on going. So we end up with the bacteria here like this, and then the cell just keeps going and starts to engulf the bacteria. And it keeps on going until these two ends meet, and these phospholipid bilayers meet like that. They'll meet round there. So yeah, we end up with a situation where we've got the bacteria. So there we've got the bacteria, but now they're fully engulfed, or almost fully engulfed in the cell, like this. And these two ends will meet, and the membranes will merge, a bit like the merging of a bubble, if you've ever blown bubbles. So what we end up with is the bacteria actually now fully inside the cell, like this. And then within the cell, there are vesicles that contain digestive enzymes, particularly lysozyme contained in these vesicles, which are lysosomes, and they move towards this. And then again, if we look at this in magnification now, if this is the vesicle which contains our bacteria, then these vesicles will merge with this, like this. And they'll just merge. And then again, this membrane here, the membrane just merge into one. And what this means is that the contents of these lysosomes, which is largely lysozyme, digestive enzyme, gets in and it physically digests the bacteria. Digests them. And it converts them into fatty acids and amino acids and other simple things like sugars, and they're simply absorbed into the cell as nutrients. So we start off with rogue bacteria in the cell or in the tissues that's phagocytosed by the phagocyte and physically digested. Quite an amazing process. And again, this phagocytic process basically works against any type of microorganism. Certainly works against bacterial infection. That's why if you've got infection in an area, you get lots of these phagocytic cells here. And some of them will die, and if there's a dead phagocytic cells and dead bacteria, that becomes pus, which is an obvious indicator of infection. So that amazing process there of phagocytosis. Now another mechanism, if we can line up, another mechanism here is complement. Now complement is a group of about 30 different proteins found in the blood in inactive form. And these complement proteins actually form a cascade. It's a bit like the blood clotting cascade. So the presence of bacteria will trigger one component of the complement cascade, and that will trigger further components of the complement cascade. And then various proteins in the complement cascade perform specific immunological functions to eradicate the bacterial infection, which has itself triggered the onset of the complement cascade. So normally the complement proteins are inactive, but they're activated by the presence of bacteria. And the activated complement proteins will perform various immune functions, like some will be inflammatory mediators, others will label antigenic organisms for destruction, a process called opsonation, and others will actually form tubes, sharp tubes like needles, that will perforate bacterial cells and cause the death of bacterial cells by physically perforating them and allow them to fill up with fluids and to lyse, to break up. That particular protein that penetrates bacterial cells is called membrane attack protein, so it's very useful. It eradicates the bacteria. And then we notice there's other these are actually antiviral mostly, these interferons. So the complement's largely antibacterial, the interferons are largely antiviral. Now, viral particles can only reproduce inside cells. So when some cells are infected by viral infections, these are intracellular parasites, they're inside the cells. So bacteria are extracellular parasites, they infect outside of the tissue cells. The viruses infect inside the cells itself. And certain cells, when they're infected with viruses, will release interferons. And once they're produced, these interferons will diffuse from the cell to the surrounding areas where they communicate with adjacent cells. And they will do that by actually fitting onto interferon receptors on adjacent cells. And this will stimulate a physiological response in the cells that have been stimulated by the interferon. And these stimulated cells can do things like producing antiviral enzymes. And as well as that, the cells that are stimulated by the interferon will be less able to produce or to synthesize any new proteins of their own. It will inhibit protein synthesis. And that's actually where interferons get their name from. They interfere with the cell's ability to produce new proteins, and therefore if the cells can't produce new proteins, they can't be hijacked to produce new viral particles. So again, the complement and the interferon are going to work against a wide range. The complement will work against a wide range of bacteria. They're not specific. The interferons will work against a wide range of viral infections. Again, they're not specific. And another cell that's particularly interesting is the NKCR cells, the natural killer cells. These are a type of lymphocyte, so-called large lymphocytes, and they will kill virally infected cells. And under the right circumstance, fortunately, they will also kill cancer cells. So the natural killer lymphocytes, the large lymphocytes, not the small B and T lymphocytes that are associated with antibody production. And actually, we could also include the lymphatic system in there, but we'll look at that when we consider normal A and P. Now, all of these are innate. We possess these as a result of our normal anatomy and physiology, and they're not specific. They work against a wide variety of potential infecting organisms. We have this immunity all the time. We don't need to acquire it. But with the acquired immunity, at one time we are not immune to something, and then we develop immunity to something. This is acquired as we go through life, but it's specific to particular infective organisms. It's acquired and it's specific. And the acquired immunity breaks down into two forms. There's active acquired immunity and there is passive acquired immunity. So active acquired specific immunity, passive acquired specific immunity. Now, the active immunity means that the individual who is enjoying the immunity has made the immune proteins, has made the antibodies, that is the immunoglobulins. So antibodies and immunoglobulins are the same thing. The person who's enjoying the immunity from the presence of these immune proteins has made the immune proteins themselves, actively. They have actively synthesized these immune proteins. And they will do that for one of two reasons. One is because they've been infected by the organism in question and they've generated a specific immune response to that organism, or alternatively they've been given dead or attenuated versions of that organism or antigenic components of that organism in the form of vaccination. And this will allow them to generate an active immunological response, generating active immune proteins, but without having the condition themselves, because the vaccine consists of dead or attenuated antigenic components from the organism. And this means basically that the immune system will recognise the organism and be able to eradicate it quickly should it come to infect their body. So whether someone's been exposed to the infection or whether they've been vaccinated, the immunity is active because they've acquired it. They never had it at one time. Then when they get the infection, when they get the vaccine, they acquire the immunity and they are actively immune, but it's specific to a particular etiological antigenic organism. An antigen being anything the body recognises as being foreign. In fact, an antigen is an antibody generating molecule. Antigens generate or stimulate the immune system to generate antibodies. Now, passive immunity is also very interesting. None of us would be here without it. You would all be dead a long time ago if it wasn't for passive immunity. And this passive immunity takes different forms. So first of all, we'll think about the transplacental and the colostrum and the milk. Now, transplacental is just what it sounds like. The ready formed immune proteins, the antibodies, are going from mum into the fetal circulation. So antibodies that the mother has actively made because she's been exposed to infections in the past will migrate across the placenta. It's not a passive process. There are specific carrier proteins in the placenta that facilitate the transport of these immunological molecules, the immunoglobulins, these immune proteins, from the mum to the baby. The mum has made them actively, but they're now in the baby, so the baby has them passively. They will still perform their immunological functions, but the baby hasn't made them. It's a passive immunity. The baby is enjoying the immunity without having to go to all the trouble to make these antibodies themselves. And this transplacental migration of antibodies occurs mostly in the third trimester of pregnancy. That's why sometimes babies that are born prematurely can have lower amounts of these immune proteins in their blood, therefore are more prone to infection. One of the reasons that premature babies are more likely to get infection than full-term babies. So, for example, antibodies to measles, rubella and tetanus diffuse quite easily from mother to baby. Polio and pertussis perhaps to a slightly lesser extent, but some immunity can still be enjoyed. And these are all what we call the IgG, immunoglobulin type G form. And they're active against viral and they're active against bacterial infection. IgG is the most common form of serum antibody, actually. About 75% of immunoglobulins are this IgG form. It's the simple Y-shaped immunoglobulin. Well, say simple, it's by no means simple, but it's just got two active arms and a body portion. So this is great because when it means when baby's born, in theory, baby can be immune to everything that mother was already immune to. And this lasts for several months, and even some effect up into for the first year of life. Now, like me, you've probably come across mothers who don't breastfeed at all, and this just grieves me. Because especially when breastfeeding starts, there's a less milky, serum-rich component called a colostrum that comes out from the breast to begin with, straight into the baby's mouth. Absolutely wonderful, the colostrum. And then the milk after that. Because both of these are conferring immunity on the baby, passive immunity on the baby. It's an absolute tragedy if they don't get it. And it's probably got lifelong effects, this immunity that comes from the mother, as well as the immediate effects that the baby enjoys in protection from infection. Colostrum and milk actually has immunoglobulin type A's, which is just a slightly more complicated immunoglobulin. And these form the first line of defence because they protect the gut in the infant. They're transferred into the gastrointestinal wall of the baby and the esophagus, the mouth, the stomach, the small intestine. And they're active against bacteria and viruses. So bacteria and viruses that the mum has come into contact with, the baby's gastrointestinal tract will enjoy immunity too. And of course, gastrointestinal infections are such a tragic infection in young children, especially in many poorer areas of the world. Now, most people know that there's a lot of immunoglobulin type A's in colostrum, or a lot of immune proteins. But fewer people understand that these carried on being secreted in the milk. over a long period of time. So the World Health Organization, I think now recommend that it's at least 2 years that mothers should breastfeed for. And this, of course, is the ideal situation, because the baby will carry on getting immunoglobulin type A's in the milk. They will enjoy passive, ongoing, reinforced passive immunity from the mother's milk via the immunoglobulin type A's over the first 2 years of their life. And of course, breastfeeding is good for absolutely everything, not least because it reduces mothers' chance of getting breast cancer. So we all know that we have to totally advocate breastfeeding. In Western countries, it doesn't go on for long enough. So the WHO is saying 2 years. In Western countries, it tends to be a matter of months. So perhaps our role is to encourage mothers to breastfeed for longer. And at the moment, you are producing immunoglobulin type A's that are going into your mucous membranes. It's estimated that 3 to 5 grams a day are going into your gastrointestinal tract now, but you're making these actively, of course, because you've now been exposed to antigens which have stimulated their production. And in you, IgAs are also they're in mucus, they're in tears, they're in saliva. They actually protect the respiratory and the gastrointestinal tract. So they're protecting against they're protecting the mucous membranes of the body against a wide variety of infections. Now, I've just mentioned injections there. I used to give quite a lot of hepatitis B antibodies to people. If they were going into a hepatitis B infected area, we used to give hepatitis B immunoglobulin. I think it was called gamma globulin. And fortunately, we very rarely do that now because we have the vaccination. And just as a historical point, I was first vaccinated against hepatitis B when it came out in 1983, I'm afraid. So these injections. The only one I tend to use these days in accident and emergency, we sometimes give people tetanus immunoglobulin. If they've been exposed to something like a human bite, for example, or a dog bite, and they're unsure about their tetanus status. So we'll give them the immunoglobulin for tetanus. We try and avoid it because it's expensive and it's quite a thick injection. And we'll give them the active immunity for tetanus at the same time, but of course in different arms. Otherwise we'll get an antibody-antigen reaction. Now, just before we finish this section, I want to talk about active on passive, because this is so important. Because when the baby is enjoying passive immunity for the first few months of life, up to the first year of life where there's still some effect, during that first year of life, they're going to be exposed to so many different types of environmental organisms, both bacteria and viral. So that's inevitable, they're going to be exposed to all these infections. But hopefully they're enjoying passive immunity to these infections, so they don't get the active infection. But they're still being exposed to the antigen. So that means they can generate their own active immunological response while enjoying passive immunity from the mother. So that means by the time a child is 1 year old, it can have developed active immunity to a massive range of environmental organisms, but never get the infection because it's covered by the mother's passive immunity. So this phenomena of active on passive immunity just shows us how important it is to get all the benefits from passive immunity that we can. And of course, we now know that the immune system is involved in all sorts of things for all of the child's life, probably protecting them against cancer in later life as well. Not to mention the massive epigenetic effects that breastfeeding has, beneficial epigenetic effects. But that's the topic of another video. So that's the basis of the immunity, this active and passive acquired and the innate, all aspects of immunity, which is the body's ability to resist infection.